Well, this has been a really excellent conference, and it's a real pleasure to be here and have an opportunity to talk about my thoughts on the evolution of cancer suppression, a topic that I have worked on and off for more than 15 years. And when I first started on this in the late 90s and published a couple of papers then, which I'll sort of refer to, even though it's sort of ancient history, it was pretty clear from comments that I got from various submissions that the cancer biology community really wasn't quite ready for evolutionary biology at that point. And I think we all really owe a debt to a lot of people who are actually present at this conference for changing that environment over that period of time. So a few years ago, I thought it was maybe appropriate to start getting back into it. And anyway, so I'll give you my take on the current situation, if I can get this to work. Okay, so adopting an evolutionary approach, as we all know, cancer research has been extremely successful in understanding the how of things. I mean, how tumor suppressor genes and the like work, but it really lacks I would argue, lacks a robust framework for understanding the why. And many people have talked about the importance of ultimate causation, which is, of course, rooted in evolution. But as a result of lacking a framework, there's really been no way to understand the patterns seen amongst different cancers, and I would argue the evolutionary approach allows for hypothesis testing. Of course, people have been looking for broad patterns for a very long time, and really in the 1950s, some astonishingly insightful things were determined. So Nordling, working with overall cancer risk, and Armitage and Dole, working with specific cancers, recognized that the age-specific incidence of cancer was consistent to a very simple model of multi-stage carcinogenesis involving about 6 or 7 somatic mutational hits during our human adulthood. So we start to begin to get a model, but then we've got not all cancers follow exactly that same path, and Knudsen, in his groundbreaking work published in 1971, proposed the 2-hit model rather than the 6 or 7 hits, in which retinoblastoma results when both copies of the tumor suppressor gene, retinoblastoma, are knocked out in a single cell in the developing embryonic retina. So is cancer due to, I guess there is. Is cancer due to 2 mutational hits or 7 or some range of numbers? But without a framework, it's really hard to evaluate this particular problem. Okay, so we've got a model then of multi-stage carcinogenesis, which is where you have somatic mutation driving the occurrence of cancer. And Pito in 1977 really summarized a problem, a logical problem that arises. And this is part of his quote, but I'll just sort of summarize it. Basically, humans are much bigger than mice, and they live a lot longer, and therefore we have massively more opportunity for somatic mutation. And therefore, given that mice do get cancer, we should die at a very early age of cancer. And so there's a disconnect there somewhere. And as he pointed out, there has to be some concomitant of our evolved ability to grow big and live for a long time that changes this argument. Okay, so this and some other reasons prompted me to start modeling this issue of cancer suppression way back when, and the introduction of the idea of Pito's paradox is something that we really had to resolve. Okay, so the resolution is pretty obvious, is that big animals don't typically get more cancers than small animals. And although we don't have a huge amount of data, it's clear that it's not the order of magnitude effect that somatic mutation would predict. And this is just some data from mice and humans, comparing them. And what you can see is that when you sort of age correct the data, the pattern of the incidence of cancer is remarkably similar. So something is happening. And of course, it doesn't take much of an evolutionary biologist to recognize what's going on, and that is adaptation. So we have a simple thought model. Animals evolve to become bigger and or longer lived, and there will be a significant increase in one or more pre-reproductive cancers because of the increased somatic mutation, results in a fitness decline if they die before reproduction. Fitness drop drives natural selection to suppress those cancers. So we have a range of potential ways that cancers can be suppressed, multiple different genes, but what actually gets spread at a particular time is going to depend on the specific genetic variants around at that time. And so what you can sort of predict is there's going to be a sort of serendipitous recruitment of cancer suppression in different tumors as they become relevant for fitness loss. Okay, so recently with a graduate student, Brian Muir, we tried actually looking at least this serendipity just to see if we could establish it a bit more firmly than it is already. And what we did was we had the hypothesis that tumor suppressor genes recruited to suppress a specific cancer should show increased expression in that tissue. And so we examined the expression of 15 tumor suppressors. We also did some proto-oncogenes I'm not going to mention, across 12 different human tissues. So we're looking at the expression of these guys. The genes were chosen because they were clearly linked to a high risk of a specific familial cancer in one of those 12 tissues. The data were from a range of studies, but they all use the same Affymetrix array. So this is before RNA-seq. But they were from different labs. And in fact, we found horrifyingly large, statistically significant lab effects. And indeed, there was substantial variation within a lab between different samples of the same tissue. I guess not very surprising with expression data, but it sort of horrified us that we were unlikely to find a pattern as a result of this. But so our method was, is the susceptible tissue in the highest ranked expression group for a particular gene of those tissues? So in other words, a gene associated with a specific tissue cancer, is it expressed more in that tissue than in other tissues? And the answer is that to a statistically significant degree, it is. There's a sort of mess. These are all the tumor suppressor genes. This is a mess on the right-hand side where it really doesn't work with this data set. But you can see that there's actually the white bars are the expression in the tissue where we expect expression to be highest, where the cancer is occurring. The gray bars is the average across all of the other tissues. And you can see that there are a substantial number, and in fact it's a highly significant result, were indeed more highly expressed in the susceptible tissue. Okay, so you can make very, very simple baseline models of the evolution of cancer suppression based on multi-stage carcinogenesis. And I did this way back in 1999, but to simplify it down to its sort of simplest form, we can say, let's suppose there's just a bunch of identical tumor suppressor genes and that there's M over 2 of them. To knock out a tumor suppressor, you have to knock out both copies, so you have M mutations that are leading to cancer in a tissue of size C stem cells. You've got K divisions during its life, and you've got a somatic mutation of U. Don't worry about it too much. The thing is, the incidence of cancer, assuming cancer is reasonably rare, can be simplified down to a nice, easy form, which I'll now go through very quickly. So we've got the fact that the risk of cancer increases linearly with body size, assuming body size is related to tissue size. The risk increases a power function of lifespan, assuming lifespan is related to the number of divisions, and it also increases a power function of somatic mutation rate. These conclusions basically just formalize Peto's paradox. Big long-lived animals have a problem, but obviously only if cancer suppression is considered to be a static property, which it clearly is not. So what can we do with this? I mean, is it useful? Well, there is one group that my heart goes out to who actually adopted this approach to try and see if they could come up with something interesting by doing a piece of comparative work. A group at Rochester, Andrei Seloanov and Vera Gorbanova, and they decided that the ideal group for doing this, looking in cell culture, was the rodents. The reason being is rodents vary enormously in size and in longevity. And the first thing they looked at was telomerase suppression. Telomerase suppression is assumed to be an anti-cancer mechanism. And what they found was, I must say, astonishingly good association between telomerase suppression and body size. And over here, the winners on this contest are the beaver and the capybara, who happen to be the two biggest rodents, hence their position there. But the nice thing is that this has to be independent evolution because capybaras are in this clade and the beaver is in this clade. So they're essentially unrelated within the rodent clade. Now, one of the things they immediately noticed, and you may have noticed from the last equation that, in fact, the relationship to longevity should be stronger. And here we have the little old naked mole rat, not very big, but lives forever. I mean, living more than 20 years and not getting cancer. So what's the deal with that? So they looked at it in more detail. And if I can find the right button, and they found yet another process going on in their cell cultures of the naked mole rat, which was, they argue, driving cancer resistance. And that is that there's an extra layer of contact inhibition driven by this high molecular mass hyaluronan, which is a polysaccharide. And in fact, they've gone on to look at another unrelated mole rat and found a different mechanism. The point being is just in this one example of looking at a clade, you get these interesting results that conform exactly to what we expect. And indeed, this finding of this particular sugar has actually already precipitated interest in whether this can actually be useful in a therapeutic context. So it's not that there's no applied aspect to this. Okay, last thing I want to go into is why does cancer persist, and trying to provide a baseline for the sort of models that Amy is presenting is under the simplest situation, what do we expect the baseline occurrence of cancer to be? If you've done any population genetics, you will have seen mutation-selection balance equation. It used to be nice and simple. Well, cancer is a multigenic trait, and don't worry about this, but just to say you can do it, you can work out the mutation-selection balance. The result of that is, of course, you expect some frequency of early onset cancer because natural selection isn't perfectly efficient. So you get a mutation selection balance, you'll get early onset cancer. You can predict what level that should be at. Okay, so then, of course, the main thing, however, as we all know, is we get late onset cancer, and we all know natural selection is ineffective during the post-reproductive period. And this sort of slightly complicated looking thing, I'll walk you through it, but basically I'm looking at a very simple life history where you have an equal pre-reproductive period and post-reproductive period, and we're comparing cancer risk in those two periods. The x-axis is actually related to size. So imagine that you're a little organism where one tumor suppressor will control a particular pre-reproductive cancer in a particular tissue, but you evolve to be bigger. As you evolve to be bigger, the risk of pre-reproductive cancer is going to go up. But you reach a point where selection is effective and you recruit a second tumor suppressor. You evolve to be bigger still, and then you recruit a third tumor suppressor, and so on. So you got this sawtooth pattern in the expected pre-reproductive frequency in cancer. That is independent of the number of tumor suppressors that you have. However, post-reproductive cancer is not. And this is a log scale. So when we're looking at this point at the end, this is 10,000 cases per million individuals in this particular simulation. And so what we end up with is a prediction that big, rapidly dividing tissues, which are the ones we expect with the most suppressors, will have the highest post-reproductive incidence, but no necessary elevation of pre-reproductive risk compared to other cancers. And human distribution is at least consistent with this, is that typically dominant cancers post-reproductively are epithelial cell carcinomas. When you look at mice, they have an entirely different distribution of cancers that they're tending to get. So it's at least consistent with this effect. And then just to summarize, so cancer suppression is an evolving trait. Simple model of multi-stage carcinogenesis, even though it's a very simple model, provides, I think, insights into patterns of cancer risk and the levels of pre- and post-reproductive cancer. The expected link with tumor suppressor gene expression and the tissue it protects was supported for the majority of the tumor suppressors we looked at with very sloppy data. So we were surprised that that worked. And medically useful data can be obtained from model-driven examination of non-human mammals. So thank you very much.